All right, we likely should slow it down a little from yesterday. I know yesterday was a little bit uh, excitable, and we kind of bounced around on topics. And really, as it comes down to the products and the science, some of that, when we do it, we have a certain goal in mind, and that is to understand the microbiome between a healthy and an unhealthy person. And then from that, you want to understand, why are there differences? Is it differences because they have genetic predispositions that drive their microbiota in different directions, which is in the case of some people, that's true. Some people's genetics attract certain organisms or fail to attract and maintain other organisms and feed and nurture them. So there's a genetic component. There's obviously a dietary component. There's obviously a toxic load component. You can alter your microbiota. That's probably why my voice is going down. I keep yelling. Yeah, my voice was a little raspy. There's a number of processes. This is a really complex problem with a really elegant solution. And because it's taken us 10 years to get to this point, we're not afraid to go another 10, 20 years to the next point. Because the amount of brilliant things that have happened to the world of humans and animals regarding the microbiota has transformed. I mean, we even do some studies on horses where they send us in their microbiota and we try to tease out what's going on. You know, there's a guy in Durango that raises thoroughbreds, and they're so interbred that they're struggling to find healthy babies anymore because they've been breeding them, you know, trying to get the strongest, fastest horse, and they've lost some of the genetic diversities. And there's a microbiome component to that. So there's a lot of these little fun side projects. But in our case, as we've mentioned, it started years ago, and it started because we were seeing results from the use of probiotics in various cases. It turns out when I was in college, Dr. Oberg, he works with Chris Hansen's, which is a probiotic company. This would have been I don't want to date myself, but this would have been in 1990. It would have been about 1997. We were putting probiotics on tampons and handing them out to people on campus to be in a study for UTIs or reoccurring yeast infections and such. And it was sponsored by one of the groups there. That's how long ago I remember probiotics being beneficial in some circumstances. And I'm sure, you know, there's times in your career you've found where you've tried a probiotic and you didn't get a result you needed, and so you had to try something different. Not unlike any other product we ever use, or any other process we use, or any other technique. However, in my world, I was seeing a lot more positive results coming out of people regarding bugs than I was seeing rejection or failure. So clearly, I was hooked from day one. I was hooked many, many, many years ago, and fun to see that it was part of it's what bridges every one of us. It bridges us. Yeah, we share a loving glance or a wave or we share a lot of things, or a hug or an embrace, but we literally share some microbiota. This weekend, some of you are taking some of our microbiota home with you. Hopefully it's a good thing. Hopefully you guys appreciate that. But we are literally sharing these genes, these traits, and a number of other things. So that being said, the product side of things has been slowly born, and I mean really slowly. It would have been nice to say, okay, we want to target the brain, we want to target metabolism, we want to target skin, etc. Why don't we just buy a bunch of probiotics, make 5 or 6 products, and sell the heck out of them? Make them good, make them be clinically studied probiotics from reputable suppliers. And this happened, by the way. These are things people did. But when we started getting into it, yeah, that's great. I still use those clinically studied probiotics in ours. But that isn't all we wanted. We wanted more. And so we did. And in fact, I have a colleague that created just that exact same scenario at NutriCorp under a brand called Solaray, and it's called My Microbiome. And I worked with him for many years. And that's all he did is he made a formula for all the body parts. He's got lots of money. It's a $500 million company. Went out, just bought all the probiotics, put them all in capsules, sold them. He was at market within 12 months of when the idea came up. That was 10 years ago, 11 years ago for us. Because we wanted to build these exotic, and when I say exotic, they're not exotic. They're in many of us. These are human-based organisms. And so we'll get into a little bit of those. And many of them you've seen or heard, and some have even made it all the way to the news, honestly. So human probiotics, the ones we're interested in, and the new term, by the way, which I'd like everybody to use because it's kind of fun and catchy, they're called next-gen probiotics, right? Next-gen prebiotics. Old prebiotics are just the fiber, psyllium husk, black walnut hulls, whatever. The next-gen prebiotics are all of these new and amazing polyphenolics, flavonoids, OPCs, urolithin A from ellagic acid, and so on. So what else we got? We got unique interactions of metabolites. This stuff's been really fun mechanistically that we'll get into a tiny bit. They're extremely difficult to culture. So when you ask yourself, why in the world would it take 10 years? I can tell you that's the answer right there. I can pull anything from nature. If you guys want me to pull a compound out of a plant or find a gene or find a bacterium in blood, we can do that all day long. We've got multiple PCR machines. We got multiple sequencers. We got microbiologists that rock my world every time. And we've got multiple chemists. We can pull heavy metals out of humates. We can pull heavy metals out of fulvates. We can isolate unique heavy metals from the soil, just even outside of our office. We know what goes on there. I can take the shirt off your back and tell you how many heavy metals are in that shirt right now. Well, okay, in an hour from now. Which there are many, by the way. We had leaded gasoline in America for a long time, so just about every polymer, cotton polymer, contains lead. Did you guys know that? I have not found a cotton polymer that does not have lead in it in the United States. Because of all leaded driving they did. And if you're Midwest area, where they stopped lead gasoline usage last, those levels are the highest, by the way. But anyway, so we have lots of fun, but guess what? This became almost an impossibility without one man. That one guy, Michael, he figured out how to recreate the human GI in a test tube. And before he did that, we had failure after failure after failure. They're so sensitive. There is one organism that when it grows on the plate, and he's got it under all the right conditions. And remember, oxygen is one of the conditions you have to completely eliminate. All the people in your gut can't stand oxygen. In fact, it kills them instantly. But he grows up a nice black colony. If that colony gets exposed to even a micromolar amount of oxygen, it turns white. It's almost like watching a movie where it goes black, just black from white, just immediately. And everything in that colony just died from oxygen. So he's figured out a way to do that. And there's multiple ways that he did it. He did it through the media, the growing process. He did it through gases. So in his world, there's all these chambers that control gas in his area. No oxygen, lots of hydrogen, helium, nitrogen. He plays with the elemental gases to make mixtures that match what happens here. Does that make sense? And it's in a sealed chamber. I go up and just put my face up against it and make fun of him. But it's all controlled. And after that, he has to use very specific ingredients. Some of those I can't tell you all of the details about them because he literally discovered certain types of carbons and certain types of silicas and certain types of essentially amino acid combinations that allow him to grow organisms that you can't get to grow outside the body very well. Now, I thought this was pretty amazing because I know there are at least 100 different researchers trying to grow these organisms, at a minimum, because they're amazing. Let's talk about their amazing. Actually, let me go forward one more. Here's a snapshot of them. Now, some of these you'll see, and remember, they're strain-specific. Sometimes you find Bacteroides or Pediococcus or an Enterococcus that you don't like. These are all the good guys. I don't know why this one shrunk over here, but that's Akkermansia. There's Eubacterium limosum, Rectali, Collinsella. There's Fragilis, Ovatus. Some of these names probably sound familiar. I'm going to be the first round of next probiotics. And no one's ever grown them. People have known about them for over a decade. But they became very difficult to culture and run safety studies on, and then track their metabolisms. What do they make? What makes them beneficial to us? And one of probably the biggest superstar in here is you got Akkermansia muciniphila. That's now a really big name in the industry. Have you guys heard of that one? That is the skinny bug, or one of the early skinny bugs that people had. That's one where they could take and put it in an obese mouse, and that mouse would lose weight. Now, we're not as simple as mice, so when they did it to humans, they didn't get the same result. However, a lot of what it does benefit our metabolism. So it's still doing an amazing job, just not black as white as you eat it, you become skinny. Didn't happen. However, there's some really cool things about it. It actually consumes mucin. So it's one of the organisms that lives on your mucus layer in your intestines. There's another one you guys are aware of called Helicobacter pylori that lives on mucin. And that one is also a commensal for many people, and there's a number of mechanisms that create the danger of that one. Because did you know that we're now adding that back to people in microbiome studies? Eradicating H. pylori has led to a lot more problems than leaving it alone and just dealing with individual people that had particular challenges. When their mucin layers shrank too far, H. pylori could sense uric acid production. It zooms into the tissue instead of staying in the mucin and starts eating the lining of the stomach instead of staying where it's supposed to. But when you remove H. pylori from all these people, they have all kinds of microbiological problems which lead to physiological. So see, we're learning how to live with these things, not learning how to always kill them. Even ones we think were scary for a while. And this is an ongoing theme that I'm sure you guys will appreciate as we go. However, circling back to these bad boys, these are my fun ones. These took a very long time, some easier than others. Prasnutzi down here, this is a good one for your brain. Produces a ton of butyrate. What is butyrate? It's a short-chain fatty acid. Also produces acetate, but it needs a friend to help him sometimes. What do you guys think butyrates do from the microbiome or microbial environment? What happens when they send those butyrates into the bloodstream? What is your first instinct to think that's going on there? I mean, where else do you guys see butyrates? Do we eat butyrates? Pardon? Yeah, butter. Short-chain fatty acids. They act like ketones, right? I mean, in fact, in the chemical sense of the word, they can do something else. And something else they do that I think is every bit as important is they become epigenetic signals. So when these organisms are sending in these butyrates or these modified acetates, we know when those circulate through the bloodstream and run into particular tissues, which they've been traced to, they can trip and turn on and off genes through histone deacylation or methylation on methyl C groups. Does that make sense? Oftentimes, these bugs are sending signals to your genes to turn things on and off. You want a new one? Thank you. Supercharged. But I sound about the same though, right? Start beeping. Next time you need that little three-dimensional battery above my head. So I want to kind of pick up the tempo because for us it was pretty exciting, but it was also exhausting. So my tempo right now, it really does reflect how long it took for us to find and identify and grow these organisms. And I know a number of laboratories, and if you guys have ever seen publications, there have been some really amazing studies done on these organisms. Here's the problem. When I reached out to the laboratories that did them, they can only grow them at low levels, meaning that they grew just enough in their lab to run the experiments. That doesn't do me any good when I want to grow this much to make capsules to go into 3,000 bottles. And it's that scale-up process that they all said, good luck with that. We grow this much. We keep it at minus 80 degrees because if we take it out of the freezer, it dies in 20 minutes. I'm like, man, those are fast experiments. You guys are like pipetting and doing, yeah. It doesn't like to live. That's how we found over a long period of time how to do that. And there's some really fun ingredients. I told you guys yesterday, glutathione is critical. Cysteine is critical. We bathe these organisms in a really interesting carbon silica mixture. When we are drying these organisms, and how we dry them is we grow up a whole bunch, and instead of, you can't apply any heat, any oxygen, any of that. So they're in a vacuum. They're in a vacuum, kind of like outer space. And then to that vacuum, you start pumping off any air left, and the temperature drops. So you're getting what's calling a lyophilization. Anybody ever done that to their fruit leather? It's a cold drying process where you're subliming the water. Water leaves the organism because of the low pressure and low temperature. You turn the water directly into a gas. And if we do that gently in the presence of some really cool compounds, those organisms come together and they're happy. And so far we've had them happy for over a year on a shelf. So they've got at least a year's shelf life. I thought that was pretty fun. Pretty cool. Have you guys ever tried to grow anything and keep it alive for a year? Besides your puppy or your kids? It's pretty dang tough to really do that. So I was pretty proud of that. Yeah. They have a hard time even keeping the wine, the recycling wine yeast mixtures alive. They have to switch those up because you can create genetic changes and things. So yeah, it's a tough process. This is, again, the same diagram. You guys have seen this many times before. I wanted to remind you that the original probiotics, the probiotics that have been on the market for anywhere from 80 to, let's say, the last decade, we've put a lot of energy into probiotic research, probiotic understanding. It mainly focuses on the lactobacilli, some Pediococcus, some Saccharomyces, and of course the bifidobacterium. Those are the main players, and bifido and lacto being by far the largest two. We've spent a lot of time deconvoluting the mechanism of how they benefit us and help us and send signals to our body. how they can colonize, how they might not stay very long, or how they might take up residence for quite a while. But with the next generation probiotics, we're learning a lot of new and more interesting things regarding where they colonize, how they colonize. Some of them seem to be communicating through the adrenergic receptors or the afferent central nervous system, some through GABA and 5-HTP or serotonin, or 5-HT, some directly through immune cells. Some immune cells are grabbing some of these organisms up and carrying them different places inside the body. So they're using immune cells to do some of their heavy lifting, and then finally you've got enteric goblet cells. You've got all these communications that can happen through those. And as you recall, your nervous system, your blood system, your immune system, everything is within one cell from all those bacteria. They're neighboring every possible freeway. They're like plop New York in the middle of the country and run every road to it. They have access to everything in the gut. Everything. There's nothing they don't have access to, which is why it's fun to find some of these organisms that start here. And for example, during pregnancy, certain macrophage will grab organisms out of the gut and carry it up to the breast tissue so that some of the breastfeeding, you're actually giving probiotics to the baby, even though they weren't there to begin with. They're delivered to the breast tissue to then be delivered to the baby. And they were pulled from the intestines. I thought that was phenomenally cool. You know, little guy down there, hey, I need to grab you and you and you because you got a job to do up here. You're going to be in the breast tissue for the next couple days. All right, let's go. You know, it's really fun to think about how this is all happening. Right, back to the bugs. We love our bugs. I should have put the name of all the scientists, but let me tell you, there's Ross, there's Michael, there's Toby, there's Stacy, there is Patty, Madeline, Derek, Mandy, June. We love June. So these are people that have all worked on this project. And some of them have PhDs, some of them have masters, and some of them have bachelors. But they are the most talented and nice people you'll ever meet. And they're passionate about what they do. If you tell them to go look for a bug, they're like giddy. You know, they're just thrilled to do some of these things. All right, so the next generation probiotics have a lot of data coming in. We know that they can contribute antioxidatively, they can contribute to anti-inflammatory. There's a number of styles, Phaceum, Rhamnosus. These are two that can actually control and increase the aminobutyric acid and dopamine levels, so that was why I chose them. Prasnutzi. This is a really big slide. Apologize, it's quite a lot, but I didn't see anything I wanted to get rid of. So Prasnutzi has been on the radar for a while, and it won't be long before you see a pharma company come out with this product. Sooner or later, they're going to come out with a form of Prasnutzi for this particular use, for something in brain health. Because every time we study it, it just gives us up new, really fun information. So it improves overall GI health through microbial diversity. It can metabolize prebiotic pectins, aminoglycans. It produces butyrate. And when combined with Bacteroides theta iota micron and Bifidobacterium adolescens, Prasnutzi acetate consumer now creates a lot more of these mucin glycosylations as well as more butyrate. So notice how he's buddying up with other people. These are little dynamics that we never really considered until recently. So when we grow these, sometimes we have to grow two people together. Do you know how hard that is? Do you know how hard it is to grow two bugs with their own mind and their own greed, and you don't want one outgrowing the other, and otherwise you get competition? It's been quite the challenge. It's been pretty fun. So long story short, if you think about what this bacterium can do, when you think about our prebiotic that you're going to see here in a minute, whether it be the Terra or the Immune or the Neuro, which we don't have here, but the Neuro, for example, we designed these prebiotics around what we saw these organisms needing. So if you look in the middle of this, they're able to exploit energy sources like arabinose, melbose, raffinose, raminose, ribose, xylose, linear alpha-1,2 branch dextrans, arabinogalactans, xylans, citric pectin or peptides. F-prasinutzi can grow on host-derived N-acetylglucose aminoglycans. Those are the sugars found in your mucin, in your mucus. There's actually a prebiotic coming out in a few months that has these glucosaminoglycans in it, because during the winter we want to encourage some of these winter bugs to grow by giving them the prebiotics. Does that make sense? So let me frame this again for you, because this is a lot. You've got the probiotic that takes all of its food and all of its different metabolites. So you've got the organism, you got what it eats, and you got what it produces. And there's some terms here. Do you guys want to know the terms? You may already. A probiotic is an organism that should give, by definition, some benefit to the human. Or if it's a cat probiotic, it's got to give it to cat. There is a prebiotic. A prebiotic, up until recently, has been defined as a food source for a probiotic. The probiotic, like a fiber or something else, must be able to consume that organism or that prebiotic and grow. That's the definition of a prebiotic. That definition has been expanded now because we're learning that all these polyphenolics, glucosamine fits, we're learning about OPCs, resveratrol. These plant-based compounds are not directly generating things like butyrates, acetates, or the known metabolites. What they're doing is they're now being converted into other active compounds that go into our bloodstream and do a different job. So we're lobbying to get these determined as prebiotics as well, because the organisms love them. When I add certain flavones and polysaccharides and oligosaccharides to the mixture, these organisms go gangbusters, make all sorts of fun things. That's a prebiotic. A symbiotic is when you take a probiotic and a prebiotic together and eat it simultaneously. And then you got the postbiotic, and that's all the stuff they make on the backside. All the stuff they poop out, that's postbiotic. And there's others that I could go on and kill you guys. It's like they keep making names, right? Scobibiotic, they keep going. It's never gonna end. Never gonna end. But as we started developing these products, we focused on these particular organisms and what they needed to survive. And why did we pick these organisms? One, because they had the most beneficial outcomes that we knew at the time. Are there new bugs coming out that are going to be pretty amazing? Yes, there are. I told you about that athletic one that Harvard is probably going to try to turn into a drug. I mean, clearly we're not going to try to do that one, but that's a pretty cool bug. There's going to be others you're going to see coming down the pipe. But for all of you and all of us in this room, there are people, and these bugs come from people clearly. How do we start to encourage the growth of these organisms within ourselves? The probiotics are great, and I love them, and there's a time and a place to use them, especially when you're trying to switch things up. Maybe you're trying to do the hormetic principle. Maybe you're trying to increase counts of certain positive bugs in your gut. You're hitting probiotics. You're trying to create diversity. However, how are you going to bring up the bugs that are not in the probiotic formulas, but they might be in your gut already? What do? How do we get them to grow to levels where they're actively metabolizing again? They're not dormant. They're not hiding. What do you recommend we do? Feed them. Fasting is a component, no doubt. Fasting is a component. What you do when you break a fast is probably the most critical time. If you do a 3-day fast, break that fast with some really important prebiotic ingredients. Even if prior to that, you were in a heavy keto or a heavy carnivore or whatever, break the fast with things the bugs need. If you don't, that whole microbiome reverts back to what it was pre-fast. And what we're looking for is we're looking for the little people. You have a gut full of organisms, right? They're everywhere. I showed you that yesterday. However, Faecalibacterium prausnitzii might be right here. Right? He's right here. He's surrounded by Escherichia, Collencella, Clostridium. He's surrounded by all these other ones. And some of these bloom based on what you're eating. And if you studied your feces very often, you start seeing patterns. I'm not recommending that you always sift through your feces. You don't have to do that. But there are patterns. Now, if you went even further and we do microbiome testing or we do stool testing, for example, you start to notice that you have, on the list of bugs, you have a high percent of some bugs, you have a medium percent of other ones, and you have a low percent of others. Where the brilliance of the microbiome testing is going to come in is, if you've got a bunch of good bugs that are sitting in the low percent, these got to be brought up here. And you might have a bunch of bad bugs that are doing all the damage because they're growing like crazy inside of you. But through diet, fasting, probiotic use, you can reverse these trends. That's why we're doing this. And at the end, you're going to have multiple tools to do this. If tool 1 doesn't work, straight out of the gate, we try tool 2. If tool 2 doesn't work, we go to tool 3. We're going to have 6 or 7 tools, and I can promise you throughout that group, you're going to be able to dial this in, because as we've tried it in the lab, we know we've covered so many of the prebiotic necessities, and we have so many of the probiotic necessities by mixing and matching. Even people with the worst GI responses, after about 2 weeks, we're starting to get better. You just dial it in, you tease it. It's a protocol. And the protocol, I wish I had, well, I do have them up here. So before we talk about the protocol, because I think the science you guys understand pretty well. Prausnitzii, major depressive disorder, these things are becoming well studied. I'm not going to bore you with all of that. But you can certainly read about it, and it's pretty amazing data coming out of these groups. Some of my favorite 2 brain authors in the bug world, though, are Dinnan and Cryan. They're out of the English area. They are brilliant, and they study the bugs of the brain like crazy. They're the ones that wrote Psychobiome and all those books. Good guys, and they're very active in this area. So we got bipolar. These are all the next generation probiotics that we're dealing with. L. farcimenus, brand new bug. Lactobacillus you think would be common, buy it. We had to grow it. We had to find it and grow it. Found it in a healthy young lady, and here we go. And it does some really amazing things. So we're looking at LPS-induced Alzheimer's model. And when you look at this, the AB plaque deposition in microbial activation were markedly reduced in the parietal cortex of the hippocampus after treatment. And it's a growing field in terms of how fast these things are coming out. You really can spend your whole weekend reading and make no progress on anything else. We're seeing a lot of it. Let's see, the Phaceum and Rhamnosus exhibit diminished oxidative markers. I already had this on there earlier. That's a double slide. So the Vaccae, sometimes microbacteriums give a bad name because they're scary, but Vaccae happens to be one of the positive ones. And in mice, it increased the level of brain chemical serotonin. And it also worked in a human study when they killed it. So there's something on the surface of this organism that triggers the benefit, because it didn't have to be alive. They gave the dead organism to the participants, and it still did its work on bowel disease and colitis. It's pretty interesting. Next. There we go. Prebiotics. Remember we talked about this briefly, and what I want to point out on these is they're all over the place. And I'm going to ask you guys to become label readers, at least over the next few months. And when I say label readers, the prebiotics that we provide have been sought out for very specific reasons, under very specific conditions, to be partnered with very specific bugs. Does that not mean you can't mix match them? So if you get a neuro or a neuro, if I were to tell you, okay, here's the immune biome bacteria, here's the immune biome prebiotic, these are buddies, these are partners. Does that mean you can't mix and match these with something else? Absolutely not. Start here though, because there is going to be a unique journey that everybody takes by introducing all of these new organisms with all these new prebiotics. When I did it, there were some days, the initial days, and I started small, start with like a third of a scoop, 1 capsule, move up to 2 capsules, bigger scoop, over the course of a week, because you do feel activity in the gut. You feel gurgling, you feel movement, you feel the peristalsis kicking in, and so on. I want that to all take place as you're rearranging the gut with the first. And then a month later, typically a month later, is when you try the second one. Terra. And then we go down the third. And how do you decide? Here's one of the catches that you might help me decide today. How do you decide where to start and where to end? Do you start with Terra? Or do you start with Immune? Or the Neuro will be out shortly. Do you start with Neuro? The question is up to you. You can start with any of the 3, and you have to rotate in at some point. I start based on what the patient is exhibiting in terms of symptoms. Are they rashy? Are they, do they have a little bit of GI issues going on and they have a massive rash? Well, I'm going to start with skin and the immune. And so on. There might be a reason, if it's strictly GI or they're having dietary challenges, you might want to start with the Terra biome and then move into the immune. If they're tired, fatigued, that's the same 2 words, cognitive decline, maybe you want to start with the neuro and then move to the immune. The really fun thing about this science is they overlap. I don't know if you guys realize that as you guys read. The probiotics that do an amazing job for your immune system also do an amazing job for your cognitive system. There's a lot of overlap, and some of the data that I present to you, it's only because that's what we've studied. If we take an organism and study it for depression, we say, oh, that organism is really good for depression. But I could take that same organism and study it for its ability to influence the immune system, and I would get a response for that. Does that make sense? Some of the work that gives these microorganisms their uniqueness, it's because that's how they've been studied. And the more you study them, the more you find they do. Is that clear? That's why they can be overlapped a lot. The brain bugs don't need to always stick with someone with a brain issue. I'm going to use neurobiome on everybody. I may not start with it, I might put it in the middle, or I may put it at the end, but I'm going to rotate every single person through every single one of these formulas over the course of about a year. And you ask yourself, well, how are you going to do that? Well, you've got we have skin. We now have, let's call it neuro, because that's what it is called. We have Terra. We have immune. Now I have 4 months of rotation, maybe a little less, depending. Here's my protocol for that so far. Well, coming down the pipe, we've got the SIBO, or dysbiosis. Got to come up with a name that won't get me in trouble, but that is a soft shell, hard shell combo that is going to be pretty amazing. Guess where this one's going to go when it's out, though? This is number 1. This goes right over here. This is where everything starts. You with me? We begin there. Otherwise, we're kind of pushing bugs around. So let's pretend this goes to 1. We have a metabolic pre and pro. That's the skinny bug people. And don't be, it's not going to turn everybody skinny. It's a metabolically healthy formula. And there's acromancy and some others that have shown in animals to do that pretty quickly, like germ-free mice, make them fat, give them this bug, they become skinny again. We are not mice. Just be careful. That's what I'm just saying. But it's a really amazing formula. I've taken a little bit of it. You're going to love it. And it's got a pre and a pro. We then have a endocrine. And if you guys have a better name for that, let me know. But that's the formula that has all the organisms that seem to be involved in our endocrine system or our hormonal system. And those are the ones that, there's a couple in there that are specific to the male and the female urogenital tract. So that one's going to be in a capsule that can be used suppositorily, a capsule that dissolves. It's not going to be one of those capsules. All these other capsules withstand the rigors of acid, and they release down here. That capsule is going to be an easy dissolver, so you can use it as a depository if you need to. So that's the endo. Then we got the performance one. And then we have the mouth, esophagus, stomach one. And I thought that was going to be a fun one. That's going to be a powder. Or maybe a chewable. You just rinse it around your mouth. It's got a lot of mouth bugs. And bugs that we know reduce the competitiveness to the gingivalis and other mouth ones. And then finally, kid. And the kid one will come with human breast milk prebiotic. This is your rotation. This becomes the protocol. You start here. You can use testing to do it. You can use, hopefully in the next few months, we'll have the microbiome testing to help you do it. You can use questionnaires, muscle testing, however you'd like to do that part. And then you start the rotation. And I would preferably, and I think Dr. Mama said this yesterday, I like cyclical mindsets. I like circadian rhythms. The microorganisms are partly responsible for some of your circadian rhythms. You know, their effect on the pineal gland is real. And so think about your patient or your body and what it does in the spring, winter, fall, and summer. Or if you're more equatorial, how do you like to get these rhythms going? Because we do better in rhythms. And we do better if we really cater to the rhythms, not fight against them. So these can be done rhythmically. So in my world, up in this latitude, I would say I would most definitely be doing the metabolic in the winter. I would be doing the endo in the winter. I would be doing Terra in the spring or fall. I would be doing skin and Mez and performance. Where am I missing one? Oh, immune. Not in the fall. I would be rotating definitely through a cyclical pattern for me. And there's enough going on here that you shouldn't see too many challenges that you won't overcome, because we want to microdose some of these prebiotics so that we get them started. And by the way, the microbiome doesn't need macro amounts of prebiotics. It just needs micro. You can get a bloom of a really good bacterium in your gut by only giving it 1 gram or 0.5 gram. So when you take a scoop of 8 grams of this, and it has multiple ingredients, as you guys will see today, don't be concerned that it doesn't have what you'd consider a dinner plate's worth of greens or something. We're not trying to drive your physiology. We're trying to drive the bug's physiology. So we're giving them everything they need in a scoop this big. And it's more about diversity. You're getting, over the course of these products, one of these days someone will sort out for me, but over the course of these products, you've got over 100 different organisms, well over 100 different probiotics in there, and 140, 150 different plant-based compounds, including mushrooms, including unique carbons and so on and so forth. It's just covering the gamut. We tried to find the most powerful pre's and pro's to put together. And then we grew them in the lab. We wanted to make sure they grew, that this bug grew on this. It was pretty fun. And they did grow. And they like to grow. And now they're our friends. Let me grab my little clickety-clickety. All right, so the herbalomic scenario, and I think somebody else mentioned this yesterday, plants are no longer going to be seen as just a toxic or non-toxic association with microorganisms. There is a deliberate and sometimes creative relationship between plant-based compounds and the microorganisms. It's no longer as simple as we used to think it would be. For example, golden seal, we thought, golden seal has the berberine, hydrostein, that kills bugs, eat that, makes your belly feel better. Certainly nowhere near that simple. Hydrostein and berberine are alkaloids, and they do affect the microbiome, but they affect it in ways that don't act like antibiotics. They act like something that we've been talking about. They shift the behavior of the microbiome by having them present. Does that make sense? It's not just a straight-up antibiotic effect. It's a rearrangement effect, re-diversifying. So there's a lot of that going on, especially if you think of all of the kinds of chemistry that our microbiome does. I mentioned that yesterday, the 3 million genes. They can do chemistry that we can't even begin to think of. And as smart as we think we are, we can't even touch it. So pomegranate extract, that's going to be in one of your formulas. This one was really fun because a lot of people, this kind of went viral. Pomegranate, ellagic acid gets converted to urolithin, and urolithin protects a number of systems in your body. And we figured out that it's the ellagic acid getting converted by the microbiome, creating urolithins, and urolithins doing all this amazing work inside the body. So it just became a really simple story to follow. Eat this plant, the bugs will turn it into this, and you now are healthy. It became a really simple story for a lot of people to tell on social media and other ways. So now, I think there was even a podcast that someone gave all about urolithin A and how it comes from pomegranate juice and all that. More and more of these stories are coming out. Whoa. More and more of those are coming out. I got to go back. Just kind of went weird on me. There we go. All right, let's see some of my babies. Come on. Yeah, I'm pressing the back button. Oh, maybe. The light's on. All right, so that's where I was. The pomegranate, every one of these and many more are in the product. I'm just trying to highlight a few of the product highlights where you got pomegranate extract, which we know gets converted into urolithins. The bacteria that does it is in the probiotic, so we're delivering you both beneficial pieces. If you take the mulberry extract, data show that increased enzyme activity such as superoxide dismutase, glutathione peroxidase, catalase, high-density lipoprotein cholesterol, these were all as a result of using this mulberry extract. And then they monitor malondialdehyde. So, and then they related some of this positive extract to the enrichment of some key bacteria. So there's some associations, maybe not complete causations, but some correlations between all of these upregulated behaviors with mulberry and these particular Bacteroides, the strains of Bacillus and Bacteroides. Akkermansia, Bacteroides, Alicobaculum, Lactobacillus, and Bacteroidales. So as you can see, the plant world and the bacterial world are coming together in some really powerful ways. You guys used to think of plants as strictly herbal medicines. I did too. This herb, echinacea, if I have a cold, Golden Seal if I have a GI issue, and so on. Name another one. I mean, what else would you guys use in the herbal world to treat a human ailment? Because I'm drawing a blank here. Garlic, oregano, etc. Each and every one of these, although there are some physiological benefits from the plants themselves, but like in garlic, for instance, garlic is chock full of these polysaccharides that get chewed up by bacteria and become amazingly healthy for some people. But you have the wrong bacteria, you get something called FODMAPs. You guys heard of that? So garlic creates FODMAPs for the people that have the wrong bacteria, but for the rest of us, it's beautiful. It's a beautiful thing. Your breath stinks for a little while, but whatever. Every single story like that exists now in the plant world. Plants aren't just for we herbalists' use in the human. They're now also necessarily used by the organisms. And that sounds a lot like about what I was saying yesterday, doesn't it? Now, can they be both? They can be both, absolutely. The plant doesn't have to always be converted by the bacterium into something new. It might go on and be useful just as it is when you ingest it. It may be left alone or it may be modified. All of the glycosylated things that we eat, and there are many, they do get deglycosylated as they go through. And an area that we won't get into today, but we should circle back at some point, especially those of you that love this topic. What happens to bile acids coming out of the gallbladder and the microbiome is a critical part of getting people better in their microbiome. There's a lot that goes wrong from a dietary standpoint, and we produce these secondary bile acids which get metabolized into tertiary and so on. There's a lot of things happening there that has a ripple effect throughout the rest of the colon. And we have to address that. There's a number of studies that I got involved in, and we have a LC mass spec so we can pull really small compounds out of blood or urine or whatever. And these people were looking for these metabolites that were dangerous. And I think, didn't Danny point one out yesterday for the keto people? There's a metabolite that's made that's cancerous. Yeah. I don't know if that's a bile acid or not, but oxidizing fat. And so these bile acids are becoming they're beautiful and they need to work, but when they go wrong and you start feeding the wrong bacteria, you get a lot of inflammatory issues. And that'll be a fun talk during the Metabo. When we come back and do Metabo later, next year maybe, something, wherever. There's a lot of good stuff going on here, guys. Sorry. These are all some of the ingredients that went in. We used Maine, cordyceps, lion's mane, blackcurrant, blueberry. We picked the ones that I knew had the most prebiotic efficacy for things like acetylcholine synthesis, presynapse, prosynapse formation, NGF, and so on, neuronal growth factor. I used a lot of the science to drive this. This is some more of the science, the vitreous vinifera, ginkgo biloba, and so on. These are some of the markers that they make. Not going to drive you crazy there. Here's some of the bugs that we have. Beautiful little guys. They're dead, by the way. As soon as I take a plate out to do something pretty with it, it's a goner. It has to stay in that anaerobic state. And as soon as you bring it out, it's a matter of time. They're being wiped out quickly by oxygen. Sad. Oh, here. No. This is an imaged group. No, they are not this colors. And when you use the scanning electron microscope to see them, the real data comes out kind of a black and white. And then the people just overlay colors over it. But do the bacterium have real colors? Yes, they do. That's real. These guys produce a lot of carotenoids. The same thing you see in melons and carrots and all that. So these guys, when I grow these, depending on the conditions, you'll see them turn dark orange. You'll see them be more yellow. I try to figure out if that means they're happy or not. Because And they make their own. It's not like we add carotenoids to the media. We add basic stuff. We add amino acids and we add other basic needs. But they crank out the carotenoids. Yeah, it's pretty cool. Yeah, I imagine if, as much as they do when they dry down, and they're still yellow when we dry them and save them for the formula, they're still bright yellow. So there's things we're getting from our probiotics that I'm not even putting on the label. There's a ton of carotenoids in there that you just be getting, it's free. It's upsell. You're getting the free carotenoids with everything. Yeah, there's a lot of other things like that these bugs do. They're just absolutely amazing. Some of the research we did. So I mentioned that I've kind of dabbled in some of the growing, and we have to do all the sequencing. We have to sequence everything. We have to we've spent a lot of time looking for antibiotic resistance genes. So I have all the data for every bug, every probiotic, by the way, that's available in the world, and other bugs that we've pulled out of people. And I have learned one thing. There isn't a single organism that we have found that doesn't have an antibiotic resistance gene. The most high-quality probiotics, the youngest little person, poo-poo, bug out of her. And I told you yesterday why that is. Antibiotics are a natural compound. Your body is being exposed to those for thousands of years. Every time you came across a penicillium or an aspergillus mold or any of that, or a bacillus spore former, they all produce these types of antibiotics, which means your cells had to figure out how to deal with it and not be killed. So that's not the scary part. What's the scary part? We take all those antibiotic resistance genes and we make sure that they're only sensitive to a certain level. If you can give a ton of antibiotic to that probiotic and it doesn't kill it, it's out. Can't use it. You have to be able to, it has to have a little bit of resistance, which is natural, but as soon as you up those levels, it's got to die. That's the definition of the safety of antibiotic resistance genes. We have to look for toxin genes. We have to run full sequencing on all the bugs, and then we have to do things like the postbiotics or metabolomics to find out what these guys are doing. So here's some really fun data by laser. So you can see your Mycobacterium, Clostridium, Eubacterium. Fragilis does a pretty good job at making acetic acid, but check out Pediococcus acetylacti. This one, acetylacti, makes 57 milligrams per mil of acetic acid. That's a lot of acetic acid for a little tiny bug. Now you guys are thinking, well, who cares? Over here we've got Pediococcus acetylacti making lactic acid. Well, we don't need to worry about that for us. But so what I'm pointing out here is, as we run each and every one of these bugs through the system, we're finding that things like Praznutzi, Fragilis, acetylactite, theta-micron, they're making really cool compounds like butyric acid, ellagic acid. Acetic acid is actually a good thing because this will help keep the competition down from something like an Escherichia coli or an Enterobacter that's trying to take up residence. When you have this consortium over here growing and spewing out all these amazing postbiotics, you're reducing the chance for the bad bugs to take hold. And the bad bugs are coming in all the time. So this is one way that you weaponize your stomach, your intestines, your colon, and so on. By having the right bugs there, they help weaponize you. So this will be ongoing. You'll see more of this. We'll figure out how much of these become epigenetic traits and so on. We did a cell line. I did both a cell line on colon site as well as what's called a peripheral blood cell immune cell line. And we did that. Here's all the bugs. And what I wanted to show is that even under stress, none of the organisms that we're growing and maintaining would kill either your immune cells in cell culture or your colon sites or intestinal sites. Does that make sense? I don't want any of these bugs to have some lethal component to them. So that's what this study is. It's essentially a cell viability assay in cell culture. So when you look for these up to the 100% mark, you don't really have a problem. We'd also played with it too. We added, in some of these areas, this guy was a crazy, but in some of these areas where it dropped a little bit, it's because we added 20 micrograms or 20 nanograms of an RNA virus. Why would we do that? We wanted to see if these bacteria help protect the cells in the presence of an RNA virus. So we injected into the system an RNA virus. These cells get really irritated. They start making a ton of cytokines. They get inflamed. You can actually see it in the microscope. And what we found is most of the bugs didn't change the trajectory. You can see you got a little bit of cell death here because of the virus. But this bug right here, the one I highlighted, this happens to be Faecalibacterium prausnitzii. He was the one organism that protected. The cells grew higher than 100% after adding the RNA virus, which should have killed them. Bacteroides didn't do it. Bacteroides is like, yeah, you're on your own cells. But Faecalibacterium prausnitzii protected the cells, so that's pretty cool. But this is more of a safety / cytokine experiment, wanting to make sure everything we're doing is safe and effective. Conclusion, these are all next-gen probiotics. You will not see them anywhere. There's only one other company right now that offers one of these organisms. And they're killing it, by the way. So you guys should help us kill it, because they came out with a single organism called Akkermansia, and they just got $110 million round of funding. We've had Akkermansia for 10 years. We just didn't put in a product yet because we were studying it. So that being said, we have 20 unique products, well characterized, fully sequenced. We know what they're doing and making and what they need to eat, because even if you take Akkermansia as a pill, they didn't think it through clearly. You still got to feed it. And if you don't feed it, it's going to disappear. That's why we're a pre and a pro company, because we want both things simultaneously happening. And depending on the expertise you guys develop in this world, I've tried starting with a little bit of pro. All right, I'm not sure what's happening, didn't notice anything. Then I would jump over to pre before I added pro back. There's ways you can manipulate in a really fun way. And you can flavor it, you can not flavor it. I should point out that these are the real deal. I'd added no sweeteners, no flavors. You do get the taste of the real stuff in here. Now, for people with really, really sensitive palates, I might have a problem, which is why in the box, when you get it in a box, I've put a recipe book in there. You can add it to juices, you can add it to this, that, the other. But I didn't want to add that stuff ahead of time, because someone like me, this tasted a little bit like a tea. When I put it in, now it thickens more than a tea because you get a lot of these oligosaccharides, but it came off kind of with a smoky tea flavor. It's very palatable, except for some. I think some people are like, this is terrible, not doing it. This one has a lot of greens in it. And I didn't play the game of little bit of greens, lots of fruits. I didn't play that game. That's an easy game to play. Athletic Greens does it. Biome does it. They all do it. That's an easy way to flavor, where you take a couple of grams of greens, 4 or 5 grams of fruits. Now, I'm not saying fruits aren't good for you either, but this tastes like greens. You will get hints of the brassica, hints of the radishes, hints of the leafies. You will get a little bit of that. In fact, someone at work said, you know what, I might just put that in hot water and call it a tea, or call it a soup. It's different. Now, there are fruits in this, but the ratios are slightly different. There's a higher ratio of greens to fruits. And when I say greens, it's all colors. It's not just greens. We have shrooms. We have not the kind of shrooms you guys like. It's actually quite comprehensive. I tried to put as many in that dense scoop as we could, because I wanted to hit the microbiome. It wasn't about just getting your salad for the day or that. I wanted to cross these, what we know are molecular barriers that the bugs need. They don't need just broccoli all the time or just kale. They have to cross over into these other compounds to diversify. Pardon? This one's Terra biome. Terra is the really greeny one because it goes with the soil-based organisms. Where do we find soil-based organisms? In the soil, and a lot of them are spore formers. In fact, all of these are spore formers. But how did humans come into contact with them on a regular basis? By getting our food out of the soil. So I picked a lot of soil-based foods here that coincides with the soil-based organisms that would go along with this. Because I want these guys to stick around a little longer. In fact, there's been a couple of recent studies that show that these soil-based organisms, where they thought they were transient. Well, sure, go ahead and eat them. It's a waste of time. You're going to eat them, and it's going to poop out in 30 days, and you're done. That turns out not to be true. They are taking up residence, and they're becoming what we'd consider semi-commensal, where they're sticking around, they're doing a job, and they're doing it well. And they can have some pretty impactful benefits in your body. No doubt. Do you guys have any questions about this? I want you to taste it. I think we brought stuff to make the tasting experience a little better, but I wouldn't mind your input on just straight up tasting it, because I liked it, but I don't know that all your patients will stay compliant. But you can hide it in anything too. It's very hideable. And as I said, we've tried to get the best of the best here. You guys have heard of fructooligosaccharides. You've maybe now heard of galacto-oligosaccharides. We're trying to get every type of really cool. So I have in this one the galacto. I also have the resistant starch. I've also got a polyphenolic blend from blueberry, dragon fruit, polyphenolic. I've got oligos so it's actually a glucomannan oligosaccharide. So there's these unique oligosaccharides that feed these bugs. And I haven't found them in other people's prebiotics. I get it backwards every time. So we are pretty proud of this because it's taken us a long time to get it all together. This one is the immune.
Go over each of those, Shayne, specifically, and tell them each what comes in each of the packages, and then what.
So what it's going to look like, by the way, here's my immune. Let's build the immune first. You can buy them one-off, but you can also buy them, ideally, you'd buy them as a package, right? Because you really have a greater benefit by using these together. Otherwise, you're potentially losing out on an opportunity to grow them. So this would be the box. This would be the immune box, right? A little saying in there. We're going to move that up here next time so it's more legible. Then you open it up, and you have the probiotic in the small pill and the prebiotic in the large. And the serving, I would start with a little chicky person that doesn't know how their gut's going to respond. Start with a smaller dose. Someone that has been on prebiotics and probiotics and is toughened it out. Start right with start up with the total dose if you'd like. You can increase the dose. Now lately, I won't tell you this quite yet, but lately I've even started switching doses. Here is the Terra. So the Terra, same idea, in a box. You've got the pre and the pro. Everybody here aware of soil-based organisms? That seems to have enough popularity. It's been around for decades, but it's all of a sudden popular, right? Anybody want to know more about them real quick? Okay, so what they are. We'd like to think of our commensal organisms as organisms that we find living inside our gut, on our skin, in our nose. And these spore formers or bacillus species have been used quite often in industrial processes. They're so hardy. They form a spore in nature, in the soil. And then when you ingest them, they become, that is when they can actually open up and start growing inside your gut. And then they produce new spores that go out and back into the soil. So you become part of the life cycle, or another animal. We thought of that as simply a life cycle phenomenon, not necessarily beneficial to the human. It only seemed beneficial to the spore former. You gave it a place to go, gave it a place to eat. It makes a bunch of spore babies, and out into the dirt they go. Turns out that's no longer true. When these various spore formers or SBOs get into your body, and they do vegetatively grow, they produce a lot of really cool compounds. And some of those compounds are antimicrobial. So they can actually help you. They can be the bully. In fact, I know people, I don't know, you can raise your hand in this room, but I've used these products when someone's got something going on in their gut and you want to bully it because you're not quite sure why it's not behaving. You can sometimes go to an SBO and it bullies that, they eat it, goes in, they're like, oh, it's feeling better. Because the bullies are getting in there and making some antibiotics that are naturally occurring antibiotics. Or they're proliferating and squeezing out a community or making things happen. Hormesis, they're switching things up. So yeah, I've personally used these like bullies. I don't know about you guys. I like them. They're not always bullies. They're fun guys too. But yeah, we've used them that way. And then the prebiotic with them, although it is a much, much more than a prebiotic, this is a way for me to not only get the prebiotic for Tara, but also to bring the first green, organic fruit drink with a lot of cool enzymes in it to this group. We do not have a green drink. We haven't ever had one. This is so this kind of does double duty. It's a buddy for this guy, but on its own, it's something I'll probably be eating every day as a good green fruit drink. Does that make sense? That we've never had. Clear as mud. So I'm excited for this for two reasons. Yeah, it does double duty because I've always wanted to just have a little bit of a green drink every day, and we've never had one. So double duty. And then forthcoming will be the Neuro, which isn't here, but the Neuro is, and you can see the colors, and I'm really excited about the colors. You guys like the color? How about the labels? And the other day, I was just experimenting with the colors because I like colors. I'm a color person too. And I realized that some of our choices, which I think were not completely random, but there's this serendipitous correlation between like some of the chakras and the colors that we chose for like brain versus gut. And because I happen to have all the bottles on my desk and then someone had a poster of all the chakras. Like, hey, did anybody do that on purpose? Because that's kind of cool. Anyway, it's kind of fun. But the Neuro, so Neuro's got some really amazing probiotics and prebiotics, but these have both been updated. We did a beta test about, 4 years ago. Anybody want to dare venture back to that? This formula, or these two formulas debuted 4 years ago as a beta test. And we had amazing reviews. I had a couple of criticisms, which I addressed later. But these are now an updated version. There are more organisms in the bug side, and there are new prebiotics on the prebiotic side. But we really have tried to encompass every bit of the science that we could regarding pre and probiotics at the moment. So I think this is a really strong product based on having the organisms that we know in the right conditions can produce neurotransmitters. In the right body, they can do a benefit to the hormone system, the endocrine system, and so on. So we are excited about this as well, and it becomes another part of the protocol. Before I get off stage, and before I talk to you guys about the blue pill, you know what? I should say something about the skin. Is everybody here familiar with the skin now? No? Yeah? This was a really fun project that we were proud of from the get-go. And although skin pigeonholes this probiotic, I don't want you guys to pigeonhole it. This can be taken anytime, any place. I take this constantly throughout the week when I'm rotating. Today I'll take 2 immune. Tomorrow I'm going to take 2 skin. Next day I'm going to take 2 neuro. skin isn't just for skin. Remember, the reason skin works is because the gut-skin connection, those are the two largest groups of immune systems, which is why when you have a gut issue, you always have a skin issue. So I meant this to be, for skin, but also to be driving inwards the health of the immune system from the skin to the gut. But something that is way more skin specific is this little beast. Have you guys tried this? So this is the living one. This has got living organisms in it. And I bought a version of this from everybody on the planet.
What's its name?
Oh, dermacolonizer, sorry. Yeah, I'm flipping it around. Can't you guys see it? Keep up. The dermacolonizer I bought globally. Everybody that said they had a probiotic cream or serum or whatever, I bought them all. And only one company tested live, and it was out of France, and you couldn't get it here. I had to buy it and get it shipped. They actually did have something living in their formula. But everybody else, when you read it, it'll say probiotic lysate, probiotic ferment, probiotic extract. That means they're taking just all the poop, right? The postbiotics from lactobacillus, bifidobacterium, whatever, and that's the ingredient they're adding. Because I couldn't find a single living one, and I tested probably 20 different formulas. Yeah, it was pretty incredible. There were some big companies that I tested too, and I was surprised that we're ISO certified lab now, so our data, Amazon comes to us to check people. We get Amazon now. When Amazon sees a sketchy thing, we get the sample because we have ISO certified people and methods, so it's pretty. Of course, that was frustrating because we got a lot of samples that we didn't want, but made people busy, right? No one wanted to be that busy. But anyway, it's sad. Sometimes marketing can get ahead of things. This is living. We love it. It's got a lot of really good anecdotal data on it. We're working towards clinical. 2022 should be the year of clinicals. I've lined up a dermatologist that wants to maybe tackle some of these, along with I don't have it up here, but you guys seen the Skin Virome? No, I thought that was an amazing fun thing. It's the phage that kills Cutibacterium acnes. Anybody know what that is? Cutibacterium acnes is the organism that is predominantly, not 100%, but predominantly responsible for the inflammation spots you get on your skin, your back. Acne. That's why we call it Cutibacterium acnes. This phage kills that bacterium. And again, I only have a few really good testimonials or anecdotals, but they were pretty amazing. Video of beginning, middle, end, watching someone's face get so much better over time. It was beautiful. And, they were happy. Face is a big deal for your psyche, really. So we're going to do some clinical work coming up. And then finally, I would hope that you guys are excited about this, because this is where I want to play. This is where I want to help you guys in marketing. And this is where our marketing team wants to help you guys in marketing. We want to create these little games that bring attention to what we're doing, correct? And then you guys can use that in your own marketing to bring attention to your clinic. And I'm open to other ideas too. But we made what's called a blue poo pill. Okay, blue poo pill. Same labeling. We've tried it, okay? And I'm telling you, this is really fun. Because I don't know, how many of you watch your every bowel movement? Do you? Like seriously, everyone, you document it. Oh, well, I mean, yeah, you look. But I'm talking document it, because you document how long it took between bowel movements. You document what shape it is. We're creating an app where you just pick the shape it was, the Bristol or whatever. Because I was completely wrong about how long I thought it took. Have you ever heard someone say, I just went and had a taco, it went right through me? They are liars. They had all that stuff built up that they would let you smell way before that taco made it through there. Yeah, that was a setup. You know, on that long car ride, like, dude, you shouldn't have given me that thing. It can take like 18 to 24 hours for this blue pill to come out, or the blue poop of the pill. Or longer, or shorter. We have a young man in the laboratory. He's not in the lab, he's in IT. He's high energy, he's all over the place, you know, and he said that his blue poo came out in 9 hours. I'm like, no freaking way. Now, he swears by it, so I made him do it again and again and again. But here's the problem, you guys. This isn't just a game. Transit time is linked to a lot of important health issues. If your transit times are too short, so let me reverse that statement. If you have things like autism, ADHD, Alzheimer's, transit times change significantly from normal. They can be really rapid or they can be really long. So transit time can be indicative of something else going wrong. So first you have to dial in and find your own transit time. You also, hopefully when we get our app ready, you can also pick what kind of poops you're having. You know, you're having the little deer poops, the pebbles like the rabbits, or are you making a big pile, you know, whatever. These are the things you can track because they're important to your health. And with the blue poop, I've always wanted to know, and I always forget. But with the blue poop pill, you start it on the app. You start it when you took it, and then when the duty happens, you stop the timer. It records it for you. You log the kind of poop you had, and then you do it again and again and again. You try to find out a pattern, and then what we want to do is take these products and see if you can alter the pattern. If you start on a prebiotic, what does that pattern do? Does it shorten your transit time? Does it change this type of feces you're having and so on? Or does a probiotic do it? Or does a probiotic make it worse? You take a probiotic and all of a sudden you're finding that you're more constipated. Very interesting. What does that mean? It does mean something. These are future things we can talk about. In fact, marketing's probably telling me that we're going to have to make videos on what the little meanings are. There's lots of them. Some of them come from Chinese medicine, ancient Chinese medicine. Some come from Ayurveda. Some come from modern medicine. So lots of fun to be had. So if you're interested, we can hook you up with some blue pills. Not today, because I've got to make them. It took me like 2 hours to make the bottles I did because I can only make them by hand. I've got to get them on the machine. They're a mess. If I'd have touched you guys, shook your hands yesterday, you guys would all be blue today. But that's the idea. So if you like that idea, and you can do the challenge, you can do it online with all your clients, you know. Say, okay, we're doing the blue pill challenge, send them all out a bottle, have them report back on the Instagram, whatever, say, hey, my poop was blue. Or why is mine kind of a turquoise green? You know, why is mine purple? Did you have beets last night? You know, there's a lot of fun things that could happen from it. But the idea is to get people focusing on gut health, microbiome health, and if it takes blue poop to do it, I'm all in. Blue is, the majority of it is spirulina, and then I have a little bit of royal blue. Yeah, it's the blue spirulina.
Yeah.
Oh, good point. Take it with food. If you take it on an empty stomach, your stomach acid can decrease the intensity of the blue. At least in my experience. I took it with some fiber. Beautiful output. When I took it just by itself, in the middle of the day, I don't know if I could even see it because I think the fiber maintained its integrity. Blue colors are pretty sensitive in nature. It's tough to keep blue colors around.
What would you consider an optimal transit time?
I don't think I know the answer to that. There are people that have postulated optimal transit times, and it really ranges from like 18 hours to 36 hours, somewhere in there. I think that's wrong. I think it's too long. Yeah.
It can be up to 72 hours.
Yeah, and I would consider that crazy. That's insanity. You can start reabsorbing toxins if you're in there for 72 hours. You're reabsorbing stuff that you wanted to get rid of before.
Can't you use this as a tool to know when you want to break your fast as well? Because
Yeah.
if you are
a great idea. That's a great idea. I never thought of that.
Repeat the
Yeah,
question.
great idea.
Shayne.
He was just saying you can use this to calculate when to break your fast, because as soon as that blue pill comes out, now you've got your mark. Now you know that that's the last thing you ate. Yeah, good idea.
Okay.
Fantastic. So this should be fun. Yeah, keep in touch with us, and I'll get lots of bottles made of blue pills, and you guys can have blue pill fun. A blue pill party. Yeah.
Oh gosh.
Maybe I'll get like
That's
a
the
blue
name of the
pill
app.
hat. Thanks, you guys.
Okay, give him a big round of applause, everybody.